Hey everyone, welcome to a very special episode of the Coral Reef Talk. I am sharing a conversation that I had with Ken from Hydrospace where we talked about PNS Probio and denitrification and a whole lot of really cool and fascinating things when it comes to the world of bacteria. Now, if you're watching this on YouTube, you're seeing the video format of this episode, but I will be posting this to the Coral Reef Talk podcast as well. So if you're interested in listening to the audio only, it's going to be posted over there. So sit back, relax, and enjoy. What's up everyone? Welcome back to the Coral Reef Talk. In today's video, I'm joined by Ken from Hydrospace LLC, and we're going to talk a little bit about denitrification and purple non-sulfur bacteria and why all that is beneficial for your aquarium. So first off, welcome Ken, welcome to the show.
Hello, thanks for having me.
Yeah, so I guess we could actually start with talking about the cycle, the nitrogen cycle. And when you're first getting started in an aquarium, you're converting ammonia to nitrite to nitrate to establish your aquarium. And then from there, you focus on the denitrification process of balancing nitrates and phosphates and managing those. So can we, or can you rather, speak a little bit about those processes?
Yeah, so the nitrogen cycle is just that, a cycle. The typical aquarist is most familiar with the middle part of the three major parts, which is nitrification. But the first part, nitrogen fixation, is where all biologically available nitrogen enters an ecosystem. If that process didn't occur, everything on Earth would eventually die out. So it's at least as important as the other processes. In that process, certain microbes called Diazotrophs take nitrogen gas, dinitrogen gas, out of the atmosphere and convert it to ammonia, which they use to build amino acids, which they use to build proteins, which they use to build their tissues. And in so doing, other consumers eat those animals or bacteria and pass that fixed nitrogen, as they call it, up the food chain. And during processes of digestion and degradation, they can be oxidized, for example, by nitrifying bacteria into ammonia, or I'm sorry, ammonifying bacteria make ammonia, and then nitrifying bacteria oxidize both ammonia and nitrite as part of their metabolic processes to obtain energy. And as you mentioned, the result of that is a waste product called nitrate, which is a highly oxidized form of nitrogen. Usually when we cycle a tank, when we cycle as what we call cycle a tank, we end up with that byproduct and very little means of eliminating it. So we have to deploy all kinds of chemical filtration, water changes, and in some cases, we promote the growth of what are called denitrifying bacteria. The bacteria I'm about to talk about and that I grow here actually perform all three of those nitrogen transformations. They're nitrogen fixers, which is particularly interesting with respect to corals, because nitrogen fixing bacteria such as these are what enable corals to be, and coral reefs to be as productive as they are in what are called oligotrophic, in other words, nutrient poor environments. And nitrification is as important as it is in our aquariums, actually plays a relatively small role on a reef. Most of that ammonia is assimilated very rapidly by other organisms, especially microbes, for growth, because there's so little fixed nitrogen in those environments. But some of that nitrate that is produced by nitrification ends up becoming denitrified by certain microbes that live in anaerobic environments on reefs and other places. By the way, nitrification, it actually removes biologically available nitrogen from the ecosystem. And even though in an aquarium, denitrification seems like this frustratingly difficult thing to accomplish, in nature, there's actually a preponderance of denitrification. And that's why so many environments like coral reefs are relatively infertile. There's just a vast amount of anaerobic areas on Earth, most of them in the ocean, but some of them on land, like in waterlogged areas like marshes and stuff, where a pretty good amount of denitrification occurs. So that biologically available nitrogen is lost from the system. And then that's why we call it a cycle. The nitrogen fixers pick that up out of the atmosphere and start the cycle all over again. So as I mentioned, these bacteria do all three of those things. They are pretty darn good nitrogen fixers. They're okay denitrifiers and nitrifiers, and they're also okay denitrifiers. So we're getting a lot of interest from people since nitrate tends to accumulate in aquariums. So these bacteria have a history in wastewater treatment. They're used for that same purpose, to get rid of nitrate and phosphate. And they've been used in aquaculture for like 4 decades. And it's just kind of starting to pick up in our industry.
Yeah, and so because they're able to process many different things, the bacteria that you're producing, they can be used in many different environments, such as live rock or sand bed. Correct?
Well, they're a little different. So I read an interesting paper about live rock, and it was an actual study. So I've been keeping all kinds of aquariums, including coral reefs, since the 1980s. And what I've always heard was that live rock is, in fact, that was the original purpose of live rock. The big deal was that it was supposed to remove nitrates, because it supposedly provided all this habitable anaerobic habitat for these bacteria. And I recently read a paper that suggests that that's not really so. Live rock, for one thing, it just has such a small surface to volume ratio. I'm imagining is why, and then it's covered by all kinds of organisms that impede passage of water in and out of the inside of the rock. Mm-hmm. So there's not a lot of transport going on. But for whatever reason, live rock is supposed to be a pretty bad habitat for these bacteria. However, like a sand bed, it would be a great one, partly because there's all that interstitial space between the sand grains or bits of gravel for water to pass in and out. You don't want it to go too quickly, because then you just it oxygenates the environment. And these bacteria, like other denitrifying bacteria, can't perform that process in an aerobic environment. So it's important that you kind of get that Goldilocks flow rate through there. Sure. So either sand, or in another really interesting paper I read, rubble works great for that. It still has, it's so long as it's like a porous material, like coral skeleton, coral reef rubble. These bacteria and other denitrifiers live inside those chunks. And even when in some of these sites that they looked at in the paper had pretty good flow rates, but these chunks are just the right size so that you have aerobic bacteria coating the outsides of these bits of gravel or rubble, and they consume all the oxygen before it penetrates deeper into the core of that particle or chunk. So they said denitrification occurs in like rubbly environments at roughly the same rate as like sand. Okay. So yeah, you can go with one or the other. People replicating a high energy environment, like for an SPS reef tank, for example, mm-hmm. Sand wouldn't necessarily wouldn't be the most appropriate substrate there. But so if like you didn't like the look, or you wanted the denitrification capacity, you could use rubble, and that would actually make more sense, not just practically, because mm-hmm. People have their pumps and it blows sand all over.
Yeah, the water flow, it's not pushing it all around. Yeah.
Yeah, but also because it's actually ecologically appropriate. That's the sort of substrate you'd find in a high energy environment where corals like Acropora and Porites
Mm,
would predominate.
very cool. So um, once we've cycled our tank and it's established and we have all the new bacteria moving around our tank and doing all its processes, um, what's some of the common ways, I guess, that we're deficient in our little closed ecosystems that we have? I mean, we have protein skimmers that take out large amounts of bacteria at a time, probably. So,
Sometimes stabilizers. Often we don't dose enough carbon, or even if we do, the Redfield ratios are off. In other words, the nitrogen to phosphorus um, ratios don't
mhm,
match up right. And you're not going to eliminate bacteria that way. Bacteria they are determined to live on Earth. There are many hundreds of thousands of if not millions of species of bacteria, most of which we have never identified. They'll always find a way. However, the microbiome of your aquarium may not be anything like one that you would find on a natural reef, because
right.
aquariums, the physical environment and especially the chemical environment is very different than from a reef. So of course that's going to promote um, different bacteria. They outcompete things like these bacteria in some cases that are actually usually quite prevalent on a natural, healthy coral reef.
Yeah, so we can jump into the purple non-sulfur bacteria. Um, and as you said earlier, it acts as somewhat of a nitrifier, but it's really a denitrifying bacteria. And so one of the questions that I got was, well, I guess we touched on this. It depends, but will having a deeper sand bed be beneficial because the anaerobic?
Well, that is an interesting segue. And so these bacteria, all purple non-sulfur bacteria are photosynthetic.
Mhm.
They're actually so they can live in aerobic and anaerobic environments, but they prefer anaerobic environments partly because they can only perform nitrogen fixation and denitrification in anaerobic environments, but also interestingly because they can only perform photosynthesis in anaerobic environments. And that kind of creates a dilemma for them. Now, they evolved like 3.5 billion years ago at a time where Earth had negligent amounts of free oxygen in the atmosphere and in the oceans. Um, and then, of course, one of the ancestors of purple non-sulfur bacteria, the cyanobacteria, or I'm sorry, one of the descendants of purple non-sulfur bacteria, which would be cyanobacteria, changed all that by um, they were the first oxygenic bacteria, phototrophs, the ones that generate oxygen as a
Okay.
byproduct of their photosynthesis. Well, anyway, these bacteria, it's called it was one of the great die-off events because a lot of oxygen is technically poisonous and it wiped out a lot of life on Earth. And these bacteria were among a few that survived that massive extinction event, and they adapted somewhat, like to this now to this day they can actually tolerate oxygen, and they'll grow as normal heterotrophs in an aerobic environment. However, they're not very competitive. Aerobes, the modern aerobes are far more competitive against them, so they like to live in the environment they kind of evolved in, which, to get back to answering that question, is an illuminated yet anaerobic environment. So how deep of a sand bed? Not necessarily very deep, because these guys will want to live a little bit under that top layer because they want to be away from the oxygen. In fact, they're motile, they have a flagellum and they swim around. They'll swim away from oxygen. However, they also want to be near the light. So they'll swim up, usually upwards, towards a light source. And then they'll find that little zone kind of at the surface of the sand bed, which may only be a couple centimeters deep. You can have a lot of benthic aerobic microbes that live on the sand, right on the sand surface, that consume all the oxygen before it gets very deep at all. We always think we need like 3-4 inches of sand, and that isn't necessarily true. You can get anaerobic zones like, half an inch of substantial. But having an anaerobic zone at all doesn't necessarily mean that you're going to have the capacity for denitrification that you'll want. So that zone may only start a few millimeters below the surface. However, you want to have enough denitrifying bacteria. Say you've got like a tank that's just loaded full of fish, and your nitrates would otherwise be at like 55 parts per million, then you might have to have a really deep sand bed. That may or may not help. It depends on there are other types of bacteria that are going to live in that environment, and they can do other horrible things, as we know, like generate hydrogen sulfide and methane and so forth. So it's not like, where I was saying, where on Earth, there's a lot of environments like that are suitable for denitrification to occur. And in an aquarium, they're a little bit more limited. People like the sand bed a lot less nowadays than they once did. In fact, a lot of people just go bare bottom. There's other ways of exporting their nitrate, but yeah, it's really not. It really doesn't pay so much to do more than. I'd put it this way: if light can't penetrate any deeper than some X amount, a number of millimeters below the surface of your sand bed, you're not necessarily promoting these bacteria, if that makes sense.
Okay. Yeah. So with the PNS bacteria, when you're adding it to your tank, then just pour it right in, right where your lights are. Like, you can add it to the sump, but it's more beneficial to add it just directly in your aquarium, because I know these guys can survive in the dark, right?
Indefinitely, yeah. Usually as chemotrophs, if they live in the dark, they like, believe it or so they like it. They're forced to have to favor an aerobic environment in the dark. They can't use photosynthesis as a source of energy. So then they like to eat like you and I, just as organo-heterotrophs, in which case you could keep them in the dark, but you'd probably want to carbon dose, in which case, by the way, they favor acetate. So vinegar would be preferential to ethanol, like vodka or any kind of sugar.
Okay.
So vinegar is ideal.
Gotcha. Yeah, I tried carbon dosing one time, and I just couldn't do it. So, but
it's key. So I've worked in a lot of LFSs selling products like no POX or however you want to pronounce that. And I've seen people say like, oh, I've got so much nitrate, so I should use a whole bunch of this product, not understanding that they're going to create a bacterial bloom that will be a bunch of aerobic bacteria. And then pretty soon their fish are at the surface gasping for air, and their corals are closed up. So these bacteria are actually beneficial in that regard in two ways. Number one, it's less risky, because instead of like adding the carbon to add with the hope that the bacteria will grow, you're just skipping all that and you're adding the bacteria, right? You have a concentrated culture of bacteria in a bottle. You can control that. You know what type of bacteria are going in and all that. And then the other thing is that these are primarily anaerobic bacteria. So one thing people in the aquaculture industry, especially on shrimp farms and stuff, one thing they love about these guys is that they'll reduce your BOD, your biological oxygen demand, which is basically all the stuff in the water that could degrade and consume oxygen. So because they're heterotrophs, they'll consume all this stuff, but they do it anaerobically. So they basically deprive all those anaerobes of a source of carbon that they would use to grow, and thereby remove all the oxygen from the water, if that makes sense. So Yeah, so it's actually safer to use than aerobic bacteria, and certainly safer than just willy-nilly throwing vodka or something like that in the water in the hope that you'll just grow some number of aerobic bacteria and hope that if you have a bloom, your skimmer will remove them quickly enough and so on.
Right.
One other thing, dosing that you kind of touched on, you can add them right to the water. If you do that, because they will be removed by the skimmer, like any bacterial plankton will, we recommend adding them in the morning. And the reason is that you can cut the skimmer off for like 12 hours, right? And the best time to do that, as we figure, would be the nighttime. And then that it has nothing to do with the bacteria necessarily, but the fact that we've had customers turn off their skimmers either during the daytime or for 24 hours or whatever. And they're they learned very quickly that their system depends on the skimmer largely for oxygenation and
Mhm.
so anyway, they had
Yeah.
issues. So what we suggest now is like just, turn off the skimmer at night, and then that'll help stabilize your pH, for example, and then it cause less fluctuation with your dissolved oxygen levels.
Okay. Yeah. And I actually have been picked up a bottle myself to try it out. And so yeah, I turned off the skimmer for the whole day, and so it was fine in my system. Um, but I've also been battling like dinoflagellates in the reef tank because I had an imbalance with my nitrates and phosphates. My nitrates was reading 0, and my phosphates were up to like 0.2 there. So, um, but you say that the PNS actually helps balance that out because it nitrifies and denitrifies, so to help bring balance back to that.
Yeah, at least theoretically. Now I'll go back to the point that these bacteria, so I'll even go back one more step and say that we developed these bacteria as a food, and anyone who uses them should consider them that, and that alone. It's merely a bonus that they perform all these different nitrogen transformations, and also that they assimilate phosphate quite well, by the way. But that's when they're alive and thriving.
Mm.
I could never promise in any given system whether or not they will thrive, because every system is different, and few systems resemble a natural coral reef in some of the most important ways with where these bacteria are concerned. So first of all, we consider them a food. They're excellent for that purpose. Now, if you're hoping that they're going to populate the tank, and they might, then you're usually going to want to provide a habitable space for them, preferably an anaerobic and illuminated space. And that could
Mhm.
be as simple as like a bio block that you throw in a refugium where it gets some light. But yeah, there's in terms of like, so yeah, so where you said like to balance the nitrogen, the important thing there is that they're diazotrophs, and this species, not just purple non-sulfur bacteria in general, but the species in Probio, Rotopseudomonas palustris, is known to live inside of corals. And in fact, they even regulate the number, their internal symbiotic populations of this particular species, along with seasonal changes of nitrogen availability. So in the seasons, I think it's summer, um, generally when nitrogen is even less available on a coral reef, then corals will take up more of this species, and instead of consuming them, they actually incorporate them into their bodies, just like they do zooxanthellae. And then they perform diazotrophy. They fix nitrogen. By the way, so two important points there. We always think that so corals, they live off of their zooxanthellae. they get a large proportion, not all, but a large proportion of their energy from zooxanthellae. Remember, zooxanthellae are algae. They can't fix nitrogen. No algae can, only this group of microbes can fix nitrogen. All algae do, like other plants, is fix carbon. So they can take CO2 from the environment and make sugars, and that, you know, they are then the coral can use to make other organic materials, but it's that algae that fixes the carbon for them. Well, so then where do zooxanthellae get fixed nitrogen? In an environment like a coral reef where it's so scarce. They get it from diazotrophs. So these bacteria live inside the coral, they fix nitrogen, and they do it in very close association with the zooxanthellae. In fact, the zooxanthellae and the coral actually give the bacteria organic material to feed on, and in addition to the protection and all that they afford. But then it's the diazotrophic bacteria that provide the nitrogen to the zooxanthellae. And by the way, while we're on the topic of nitrate, we always think that nitrate is going to feed our corals. Nitrate is not the preferred source of nitrogen for zooxanthellae. It's ammonia. So they have to go through a lot of trouble to get ammonia if you just have nitrate in your water. They sometimes will actually have nitrifying bacteria living inside of them. And get this, so they have to have, so they take the nitrate, you have denitrifying bacteria. So you have the nitrate, you have denitrifying bacteria that make nitrogen gas, but before it's released out into the environment, then the nitrogen fixers take that dinitrogen gas and then make ammonia out of it. So it's kind of a weird, you know, backwards process when we assume that, you know, the nitrate is going to feed corals. It's a very inefficient way of doing it. And also, the worst part is that it promotes algal growth. So with respect to nitrate, again, you don't see that much nitrate on a coral reef. And that's the reason they're coral reefs. They wouldn't be coral reefs if there was a bunch of nitrate. They would be algal turf and kelp forests. Corals necessarily, even though they would like to have that nitrogen, they necessarily live in a nitrogen poor environment because they cannot compete with benthic algae. They don't grow as fast as benthic algae. So what happens is when we have a bunch of algae growing in the tank, because we have a lot of nitrates, those algae not only overgrow and smother the coral, but it's not as well known amongst aquarists that there's another thing happening. Algae release exudates. They exude these substances. They're organic substances, and they're like junk food to bacteria. A lot of bacteria feed on it. It's the same bacteria that feed on the normal sources of carbon in a reef, like coral mucus. However, they behave differently when they're given what biologists call it. It's like a junk food. These algal substances that algae leach into the water, and they grow rapidly. These bacteria, these heterotrophs that are aerobes, they consume all the oxygen locally around the surface of just around the coral, and they actually create a hypoxic zone around the coral, just where the algae is growing, and it can actually harm the coral. And they think that that's partly where coral diseases start from, is the stress. So nitrate is a really bad thing, even indirectly, right?
Mm-hmm.
And corals
so that's why you can.
They have to live in a really nutrient poor environment, so that they can. So refugiums are a good thing, for example. But if we over rely on Ketomorpha or especially as we learned in the past, Caulerpa, for nitrate export, then other bad things happen, partly because of the sorts of chemicals, the substances that these algae release around the coral. It's well known to coral reef biologists, but aquarists kind of underestimate the harm they're doing to their aquariums by allowing all this algae to grow, because it is not natural. The corals are not equipped to compete with the algae, nor to sustain the damage that's caused these chemicals, which, by the way, sometimes were developed by the algae to fight other organisms like corals. They're by design, they're noxious. But again, yeah, that's just another bad consequence of having too much nitrate.
Yeah, so that's why you can see whenever you have like green hair algae spreading around your tank and stuff, you can see corals starting to look bad before they even get overgrown by algae.
Yeah.
Yeah.
The water turns yellow.
Mm-hmm.
And carbon, there are all kinds of means of getting rid of that. The obvious, easy one, I even promote water changes over my bacteria, because water changes are the surest, cheapest, easiest, safest way to bring yourself back to that original ideal, all the a set of water parameters.
Yeah.
But I think a lot of people, number one, that gets costly depending on their availability of RO, DI water. And also, then you're depriving yourself of that thing that originally wanted these bacteria to be, which is a coral food, a natural, live, non-polluting, incredibly nutritious coral food. So it's a matter of using the two hands.
Well, I was going to say it's really interesting to me that that corals actually prefer this PNS bacteria, and they attract them in, right? So I find that fascinating. Yeah.
Yeah. Well, the actual group, so Rotopseudomonas palustris, the species that's in Probio, that's a purple non-sulfur bacteria, and it's also a rhizobial bacteria. So not all rhizobial bacteria are purple non-sulfurs, and not all purple non-sulfurs are rhizobia. This one just happens to straddle, and it's the rhizobial it appear to be enticing with the, as you alluded to, the certain substances that they secrete in their mucus to attract these corals. So rhizobial bacteria, the word rhizobia comes from root. We know most of us are familiar with like legumes, and so peas, those beans, the plants that form root nodules that host this same type of bacteria as Rotopseudomonas, the Bradyrhizobial bacteriaceae, something like that. Anyway, they're the nitrogen fixers that live in root nodules of plants. And anyway, corals are basically doing the same thing. They're creating a really hospitable environment for this particular type of bacteria, the rhizobial bacteria, that benefit them by benefiting their zooxanthellae by providing them with fixed nitrogen in the form of ammonia, which again zooxanthellae prefer. They're actually kind of janitors too, because these bacteria, their food source is the actual waste products of the coral and the zooxanthellae.
Yeah, that's fascinating. That's really cool, and I'm surprised that up till now we haven't really heard much of purple non-sulfur bacteria in the hobby. It's been in other forms of aquaculture and stuff, but not like in the hobby. But
not explicitly. So one thing we see a lot, even to this day, and I kind of lament it, and I think a lot of hobbyists are starting to as well. Too many products out there, too many manufacturers won't say what's in their products, and they'll just make a claim as to what they do, but they don't say what's in them, right? Hopefully that'll change. We don't know if I don't even know if other people in my industry have been slipping these bacteria in and just say, oh, they consume detritus, or they reduce nitrate. I don't know, but
yeah, that's another thing that I liked about your product, is that it listed what bacteria was on it, and that's very good.
Yeah, so
can research it and actually find out what it is and what it's actually going to do. And
Yeah. Well, the most important thing there too, well, number one, when you make a claim, then the hobbyist, they don't have to be microbiologists to just Google the name of that bacteria and read scientific literature that conclusively backs those claims or blows them apart. So it's nice when people can go on and actually do the research themselves, or at least just check out the claims that people make. But then the other thing too is with the advent of some of the um, analytical capabilities we now have via people like aquabiomics, we can actually target certain types of bacteria, or or thereby, we're really targeting certain conditions in our tank, but we can actually rate we can use that as a metric for the health of our tank, as a measure of how quote unquote natural our mini reef is, and so on. So it would be really nice if people started listing what species of bacteria were actually in their products. Not only that, but how they're cultured, because sometimes that determines exactly how the bacteria will behave right out of the bottle, even the same strain.
yeah, absolutely. And you guys are very transparent with the research and the papers. And I love how you can go to your website and look at the blog posts and just read up all the information about this bacteria. So that's very helpful. One question I did get from Instagram was from Spidey Bry, and he was asking, when are we getting PNS cubes or dry PNS cubes for bigger batches at home?
Okay. I've been looking into that actually. I know where he got that too. So a little background there. That conversation came up in another interview a couple months ago, and I think a lot of people are kind of interested in kind of a do-it-yourself kit. So um, the reason there, number one, it'd be less costly, and I think a lot of people too just would find it'd be really satisfying to grow these bacteria the way it is to grow phytoplankton or something, partly because you can actually see them. They produce these beautiful pigments. I should go around and actually show our grow up a little bit here in a bit.
Okay.
Because taking one step back, we raise our back, and this is actually to address the comment I made earlier that the bacteria behave differently based on the medium that they're grown in. One of the ways we grow, so Probio, for example, is grown on a tea of an aquatic plant, and that makes it adapted, at least out of the bottle, that first generation. They'll change depending on the environment they go to as they continue to reproduce, but out of the bottle, that first generation is adapted to consume plant material, which is, as it happens, like cellulose and lignin, things like, well, the cell wall of any plant or algae, and few organisms can degrade that. And that's kind of where a lot of our detritus comes from, partly because it's so poorly degradable. So anyway, Probio is good for that, but then we have Substrate Sauce, which is grown on a mineral medium. Basically, just all these different salts and stuff. It's not an organic; it's all inorganic nitrogen. It's ammonia, and it's a ammonium bicarbonate is the nitrogen source, and acetate is the carbon source. It's simpler, and it's potassium phosphate. It's just phosphate source. But anyway, these are just like salts basically. And this is something that, well, the purpose of that is, again, so that product is adapted. It's specifically used for cycling a tank. So we wanted those bacteria. We didn't want this nitrogen, because that's not something you want to do in it. You're trying to get rid of nitrogen, right? In a cycling tank, you put ammonia in there, and then you want the bacteria to eat it. You don't want anything from ammonia. You don't even have corals in there that would benefit from that anyway. So anyway, in the presence of ammonia, bacteria actually have what's called ammonia shut off. What that does is if there's ammonia in them, they don't fix nitrogen, right? So because it's energetically costly for them. You're not going to go up if there's a hot pizza laying on your table, you're not going to like just wander in your kitchen and then cook a pizza from scratch, right? It makes sense. Well, that's what these bacteria are doing. They're like, I have nitrate, I have nitrite, I have ammonia in the environment. I'm just going to use that. So it shuts off that machinery, and that's one of the things that makes Substrate Sauce adapted to cycling, among other things. But because that's a mineral medium, that's something that we can just sell dried in a bag, mixed, pre-mixed. And you can sell like one of these cubes along with it. They're just collapsible polyethylene cubitainers that as you drain them, they collapse so they don't draw in oxygen or contaminants. And they're just nice because they're clear, so you can just put them up on a light rack or whatever, illuminate them easily from any side. And so basically that's where that came from. Some people in another conversation, in another interview, were like, hey, in fact, I think they did a poll and most of the people said, yeah, that would be awesome. So we have considered that. We are looking into it. I've even like tried to figure out pricing and stuff like that. It's just that coming up on the holidays here, I've been swamped just keeping up with production of my existing products at a time when we're also developing a new product that I could talk about in a bit too, if you're interested. But I should actually just kind of show you what we've got if you're interested. And
yeah, absolutely. Yeah, take us on a tour.
Yeah, so it'd be nice if I could figure out how to flip it, but I'll just kind of go like this way. So this is actually the new bacteria. It's kind of more brownish. They're all carotenoids, by the way. It's one of the things that these bacteria is so nutritious is they're just loaded with carotenoids, and that's what you see. That's one of the things that makes them really fun to grow. This is Rotobacter, and this is one that we don't have on the market yet. This is only freshwater. So this will be a freshwater product, probably more for like shrimp and planted aquarium people. It's outside the context of this conversation, so I won't get into that too much, but this bacteria really benefits those types of systems and those types of organisms. And these two look more similar, even though they look very different physically. Trying to finagle. So this one is Rotopseudomonas, the same one that's in Probio, though it's the mineral medium. So it looks very different. It's much more red. It's like blood red. The same as Rotospirillum, which is the second bacteria that you find in Substrate Sauce, the cycling product. These are both, these are right now, these are grown in freshwater media, but they can be grown in salt water. That's one of the benefits of Substrate Sauce is there's a shorter lag time when you're using them specifically for a saltwater tank. They're already cultured in salt water, so you just throw them in, they just start growing. They don't have to go through that phase of adaptation. Okay. So yeah, the colors, so this is kind of like when they're first starting out. I don't know if you can really see the colors. A little bit, I guess. A little bit. Yeah, so that pinkish, that's the Rotobacter, the new one. Okay. I guess it's pinkish to my eye. It looks a little orange or something in the, and then this is like just kind of what the mineral media look like when they're just hydrated. Maybe if you can see it there, it's a little bit of green, but the green is actually from the B vitamins. Otherwise, it's completely colorless, and then you end up with like, either the blood red or the orange carotenoids, depending on the species. So yeah, I'm sure it's, it would be really interesting for these people to grow that stuff at home. So to Spidey Bry, I would just say, keep watching. It's good to hear the encouragement and to know people still are thinking about that and want to do it. I thought I'd show these just because these are the ones that are actually for Substrate Sauce. If they show up, it's hard to get the colors in because they're actually more of like a purpley magenta color.
Yeah, it's a little brighter than Probio.
Definitely, yeah. So some of the colors you see in Probio are from the tea. So
Okay.
there's a natural kind of orangeness to the tea that they don't fully consume. There are some things like byproducts left or whatever. But I guess sticking to the subject of of nitrate, if we're talking about the two products, both of them are actually capable of performing denitrification um, right out of the bottle. But I suppose substrate sauce for people that are starting a tank anyway, that's an important feature of this bacterium is that, and the way it's raised too, is that when you use them with nitrifying bacteria, they kind of complement each other, right? The couple important ways are that they occupy different niches in your aquarium. Um, Number one, they use different carbon sources. Nitrifying bacteria are chemolithotrophs. They are autotrophic. They only utilize inorganic sources of carbon, right? So carbon dioxide, kind of like a plant, right?
Mm-hmm.
They don't consume organic matter. So people, the dead shrimp method of seeding a tank.
Cycle. Yeah.
Yeah, I'll go on Reef to Reef and I'll see all these people that have been keeping reefs since, like me, since the 80s. They're like, oh, I've been keeping reefs longer than you, and I cycled all my tanks. So I'm like, that's great. It works, but does it work as well? I mean, it basically what you're doing is encouraging the growth of all these gnarly, sometimes really bad heterotrophic bacteria that break all that down. Then they produce the CO2, which is also going to be dissolved in the water anyway to some extent. But the nitrifying bacteria, like if you put in some turbo start or something like that, they're only utilizing the carbon dioxide as a source of carbon, period, because that's all they can do. So these purple non-sulfur bacteria, they can be autotrophic, they can use CO2, but they prefer organic sources of carbon. So that's one thing that really sets them apart. And they're not as bad in the tank per se. Like so you throw in a dead shrimp, you don't know if you're encouraging, say, for example, Vibrio to grow, something you sure as hell don't want growing in your tank right before you start adding corals.
Yeah.
So these bacteria, you're going to, like when you're cycling with them even, and using like substrate sauce, you're adding something that will, you already have the capacity to remove large amounts of dissolved organics. Will you be using an activated carbon or something like that anyway? Sure. But if you do, and I would encourage you to do that because it works really well, but these bacteria will actually prolong the life of your expensive granulated carbon media, right? So, which is kind of nice. So that's one thing that's different from nitrifying bacteria. The other thing, or one other thing, is that these guys are primarily anaerobic. They prefer anaerobic environments, whereas we all know nitrifying bacteria very much prefer an aerobic environment. So they don't even live in the same space. They don't compete at all with each other, even for that most basic resource space. They live in totally different areas. And then thirdly, nitrifying bacteria, a lot of them don't like light, and in fact are even inhibited by light. Whereas these guys, of course, being phototrophs, love light. So that's another thing. You know, they won't necessarily grow in a dark, aerated biofilter. Say if you have a wet dry or something like that, they're going to live in parts of the sand bed that nitrifying bacteria definitely can't live in. And in some ways, they actually trade. There's an exchange of resources between the two. What's the waste product for one is a food source for the other. So they work really well together. But in the context of nitrate, which we're talking about today, the one thing nitrifying bacteria cannot do is get rid of nitrate. In fact, they're nitrate factories.
Yeah.
We used to have like wet dry filters, and all they did was create so much nitrate. Because, and we would they're so good at it. They're slow growing. They're notoriously slow growing nitrifying bacteria by and large. But they don't use that nitrogen for growth. They're using it for as a metabolic process. They obtain energy doing oxidizing ammonia and nitrite. And they just do it so well that a lot of other bacteria in the tank, at the expense of these bacteria, those other bacteria would have been assimilating it and then getting exported out of, say, a protein skimmer. But now you have a biofilter, like, especially like we used to have, they were the worst, like those wet dry filters that would just, we all call them nitrate factories, right?
Right.
Well, that's what you get. You cycle a tank, and then for some reason now, that's what people think is like some kind of like mark of success. Like, oh, I got a bunch of nitrate. Well, then your tank isn't cycled, because again, as we pointed out at the beginning of this, the nitrogen cycle is just that. It's a cycle. You need something, you need a denitrifying organism in there.
Yes.
So that's the final and most important difference between the bacteria in substrate sauce and the nitrifying bacteria, say in TurboStart or whatever, is that these guys will actually then take that nitrate and start to drive those concentrations down. What I'd like to see in the hobby is that last step where we see the ammonia go up and then down as nitrite goes up and then nitrite goes down. Well, we should have that last step where nitrate goes up and then nitrate starts to go down. Now, I always temper everyone's expectations with these bacteria. How well they perform denitrification depends on specifically, not just that they can survive in the tank as it is, but also more specifically that they have an appropriate anaerobic habitat to grow in, and in a large enough one too, so that they don't just persist, but they persist in a large enough population that they can manage the input of nitrate as it's produced, so that you don't have an accumulation. One thing that they will do if they persist in the system is reduce nitrate, but we don't always know how far that will be. It depends. So with substrate sauce, I more aggressively suggest applying that directly to a substrate, obviously. So we say, instead of just putting it into the aquarium, like we were saying with Probio, when you're just kind of regularly feeding with it, with substrate sauce, the best thing to do is actually pour it onto like a block of Marine Pure or that Brightwell product. I've heard people had really good success with these bacteria.
Okay.
And if you really want to go the extra mile, seal that in a container, like just a Tupperware container or whatever, to keep it anaerobic, and illuminate it, put it under light. If you really want to go the extra mile. And it's really, it's some people might be like, that's a lot of work. Well, look how much work we put into our biofilters, and frankly, how much money we spend on them too.
Yeah.
So all we're doing, but with the biofilter, really speeding up the process of cycling. I mean, eventually, if you run a tank long enough, it will be cycled, but you have to take more aggressive steps to ensure that denitrification will occur at a rate that even comes close to the production of nitrate in your system. So, it's for what the reward greatly outweighs the effort in that case. So it's just a matter of, just pouring it into, like, another thing you could use is a big sponge, like a filter sponge or something. Chuck that in the sump, throw a light on it, or better yet, if you've got a refugium, that's a perfect place because you have the light. And voila, it's really that. This is so much less complicated than some of the old. Do you remember some of the old denitrification filters?
I mean, I've had experience with like a DIY denitrification product. We took like a PVC tube, sealed it both sides, put like bio balls and stuff in it, and yeah, had water go through it and had it slowly drip out and stuff like that. Yeah.
How well did that work?
It worked pretty good. My friend was using it on his tank, and it was keeping nitrates down really good.
Oh, wow. Do
Yeah.
you remember kind of what the concentration, the change in concentration?
You know what? I'm not entirely sure. I just know that he had like a slow drip rate at the end, so that it created that anaerobic, hypoxic like state inside, like no oxygen, whatever, and it's
Mhm.
in. So, but it seemed to work really well. His reef tank was looking amazing. So,
Cool. Yeah. I mean, you can definitely like get that um, process going with like you guys did with like an actual material, some kind of a device or
mhm.
whatever, like a filter component, or a media reactor, we might say now. But one difference between, so there are a lot of denitrifying bacteria in nature. There's true denitrification, and then there are other ways that uh, bacteria can reduce nitrate. By reduce, I mean the opposite of oxidize. They
Okay.
actually, so they reduce nitrate into nitrogen gas. Basically, they're a whole bunch of them they're all reductases. They're called. So it's not just a nitrate to nitrogen gas. Some of them will go nitrite, and then some of them, there's some weird intermediary steps like nitrous oxide and nitric oxide, depending on the organism and the environment. But yeah, they all use these reductases, and you can, without going the extra mile and really trying to, you're going to have some denitrification occur in your tank, just because those microbes will probably be introduced, and especially because they're so specialized, they'll find a niche somewhere in the tank, or even if they don't have a large population, it's a pretty stable one because they don't have to endure a lot of competitive pressure from other microbes in that specific little area they're at. Um, but uh, but to get really good denitrification rates, people always have to. Now, this is more back in the day where people had a preponderance of fish in their tanks. That might have been partly responsible for that kind of need, but
Okay.
um, we would have ridiculous nitrate levels, and then people would install like these denitrification filters. And one of the things, well, two of the things that kind of sucked about them was that you had to add a carbon source, because I think they're all heterotrophic, but I don't quote me on that. But most of these denitrifying bacteria are heterotrophs, if not obligate heterotrophs. And and then also you had to control the flow rate very carefully, because a lot of them are obligate anaerobes, so oxygen would kill them. So one nice thing about these bacteria is that they tolerate oxygen quite well. In fact, they'll grow in aerobic conditions. So you don't need as complicated of a like a filter, if you want to call it that, or some kind of device or packed bed or something. You can just like soak it into like a filter sponge, or just a huge block of ceramic material, anything porous like that, where you can have an inner core that's anaerobic, and just throw that anywhere. And different conditions come and go, as you do water changes or move
Mhm.
things around. Just by disturbing it, you probably won't kill these bacteria,
Okay.
most if they get exposed to oxygen. So, again, you don't have to play with flow rates. There's really nothing to do, like if you just chuck a filter sponge into your sump or refugium or something. So that's kind of one benefit for these guys when you're specifically, you're using them to specifically target nitrate.
Gotcha. That's really cool. You can just take that filter sponge, like you said, put it in your sump, and then, of course, you have a light that's helping your Cato grow down there. So they're going to photosynthesize with that light, right? So it's
The other nice thing too, actually, say you're using Probio, and those bacteria are raised in like a tea. The
mhm.
plant we use actually has a bunch of allelopathic chemicals that they target cyanobacteria. That's what this plant releases to kill cyanobacteria that would otherwise block the light in their natural environment. So that's how they kill all the plankton. Well, cyanobacteria, again, are the descendants of these bacteria. They're a little more complicated, and they have what's called photosystem 2, and that's what this allelopathic chemical interrupts.
Mm.
Purple non-sulfurs are immune to that. It doesn't hurt them. And in fact, they can feed on it. So that's what we use, are these polyphenols to grow them out. So that's what they're actually genetic, because they have all these capabilities, but they don't exercise them all at once. They really can't. They have to be, depending on their environment, different genes get shut off or on. And that's kind of why it's important to culture them for a specific purpose. So anyway, Probio is raised in that tea. And if you say you put them into a refugium, well, then these bacteria are already going to be adapted to consuming all of the yellowing compounds, for example, that macroalgae give off, that, as it happens, also irritate corals, especially like calerpsin. And
Mm-hmm.
we're starting to see calerpa out there again more than I have in previous years. So these bacteria like this might get more important again, or into the future, as people start using more calerpa in their refugia, because those compounds are actually, as a lot of people found in the past, they really irritate corals, and I think that's what they're really designed to do, repel herbivores, but also ward off competitors like corals and other things that might grow next to them. So anyway, yeah, these bacteria, if you soak like a sponge and throw it in your refugium, it's a good resource. It has all these resources for the bacteria, but as it happens, the carbon source that they're utilizing is actually a hazardous waste product that is being produced by your macroalgae. So
Wow. So yeah, these guys have a lot of uses in the reef tank. And you mentioned time for them to adapt, because Probio is grown in freshwater,
mhm.
and we're adding it to saltwater. So what is that length of time for them to adapt and start working or attract into a coral?
Very roughly about 3 days,
Okay.
depending on a lot of things like temperature. Temperature is a big one, and then the actual salinity of the tank. They don't like salinity so much. They'll survive in hypersaline. They'll live in environments that are so salty corals can't live in them. But they don't like it. They're not as competitive against other microbes. So they do grow more slowly. It exerts a lot of osmotic pressure on them, for example. But they adapt. It's called the leg period, and yeah,
Okay.
the overall time is usually from fresh to salt is something like 11 days,
Okay. Very cool.
something like 8 days when we brew it right in fresh water, because
Hmm.
probiotics brewed in fresh water, as I think I mentioned. But it's usually about 8 days, 9 days till you start to really notice the biofilm on the. So you don't see that in the bottle. They don't produce those films anymore. So the daughter cells are the young ones, and they're the motile ones that swim around. And then at some point, some of them will attach to a substrate and produce biofilms. And those the biofilms, once they get involved in a biofilm, then they don't become motile anymore. They can't swim around anymore. They lose their flagellum and just get stuck in that biofilm. And anyway, so you don't see much of the biofilm in the bottles, unless there's like sometimes there's a sediment in the bottom, but that's what comes off the sides of the containers. But anyway, we
Okay.
start to see that after about 8 days, and it's like kind of a reddish purple burgundy film.
Okay. Very cool. And
You
so,
might, by the way, so if
mhm.
you're kind of looking to see if it's growing, sometimes you'll see it growing right under the surface of your sand bed, kind of along the glass.
Okay.
It's just the only place you'd be able to see it, but in an anaerobic area, but if you look deeper down the sand bed, you might see a little band of orangish red to reddish purple, kind of, just like a fraction of an inch below the surface, but a really distinct zone that's colored with that fairly distinctive carotenoid that
Okay, very cool. Yeah, I'll be on the lookout for that. See if it's developing in the tank. So very cool. So you have PNS Probio. That's more of a coral food, or the use is for coral food, and then you have the substrate sauce that you mentioned that is for starting up a tank. You have both strands of bacteria in that. And then you have another product that I guess we'll briefly touch on real quick, but that's the yellow snow, and that's to like simulate marine snow within the aquarium. So it's another like food source for coral food, right?
It's kind of a whole tank food, really.
Okay.
So it's got particles that are large.
Mhm.
Snow. That's the whole benefit of marine snow in nature and in an aquarium. It's nutritious because it's loaded with bacteria. And by the way, bacteria are a coral's primary planktonic food source. But a lot of them only capture, they only can only exploit yellow or marine snow as a food source because they're not
Mhm.
capable of pulling out little picoplankton, right? They're like sometimes as little as a micron in length. So marine snow is really important in an environment for that reason, but also because it's just laden with bacteria, which is a source of B vitamins in the wild. Algae, like phytoplankton, aren't very good at producing B vitamins.
Mhm.
So, and particularly B12, cobalamin, which both zooxanthellae and corals require to survive, and neither of which can synthesize by themselves, right? And so B vitamins are generally, sometimes yeast, but there isn't a lot of yeast in the marine environment. So bacteria are the primary source of B vitamins, and especially cobalamin in those environments. Well, anyway, marine snow would be useless if it didn't have all of the bacteria growing on it. That's essentially what it is a flocculant of a bunch of bacteria. That's what makes it nutritious. So yellow snow is basically, yeah, it has those big particles, but it's everything down to like the tiniest little particles too. So so it's corals will eat it for sure and benefit from it, but then even copepods, especially your copepod nuclei, which generally you're not going to have a good population. So almost all species of copepod, that their larvae are they live pelagic. They have a pelagic life stage. If they don't have food at that stage, they're not going to make it to other stages. And in a lot of cases, they just starve in aquariums, and that limits the population of copepods that you can have, is the amount of food, especially nutritious food that's available to them. People talk, oh, they'll eat detritus. Well, sure, to some extent, but detritus is nutritionally bereft. It's detritus for a reason. It's been
Right.
eaten and eaten so many times over. There's almost nothing left of any nutrition in it. There's no fatty acids. There are no vitamins. So you can't depend on that for copepods. Well, lastly, and not the least, even these bacteria benefit from the cobalamin in particular. So they, particularly in anaerobic environments, so they have a metabolic pathway for synthesizing their own cobalamin in an aerobic environment, but not in an anaerobic environment. And it's funny, they're actually closely related to Nitrobacter. That
Yeah.
sounds
Yeah.
yeah. So it's a common component of cycling products, right? It's the nitrite oxidizer in those products. And so Nitrobacter depends on cobalamin to survive. Who knows? Like, when we see like, oh, my cycle is stalled, as people call it, or whatever, like they'll get like a weird amount of nitrite, a huge nitrite surge. Who knows? Maybe their aquarium is deficient in B12, and no matter how much Nitrobacter, you could pour a gallon of of turbo start in there, but nothing's going to happen because Nitrobacter can't survive in those conditions. So anyway, that's the big plus to yellow snow, is that it is a it's big. You could use all kinds of coral foods, frankly, to feed your coral. But
Yeah.
this one is unique in that, for one, it's actually cultured on live bacteria. Here's an example over here in these carboys. There we go. So it's just raised anaerobically like that, and then we pasteurize it midway through to stop the degradation of the particles. Otherwise, they would just eat it all. So it's an ideal stage where they're coated with bacteria and all that good stuff, and the particles are still nice and big and flaky, and the bacteria produced enough of their exopolymers, the stuff that they make their biofilm with, but it also makes marine snow. That's what makes marine snow stick together. It's rich in that stuff. So corals, I mean, they're not stupid. They have a sense of taste. Like, people always talk about, oh, they eat fish poop. No, they don't. Have you ever seen a coral eat a piece of fish poop? Because I've seen lots of fish poop on lots of corals, and it falls right off, right? Because they don't want to eat it. It's useless to them. They're not going to waste energy eating something that has no nutrition. They know what they're eating. Just like we were saying, they actually select for specific types of bacteria. They know what they're doing. They know what they're eating, and they know how to select for it. So with this product, there are lots of products that are called marine snow and cool, but this one is actually made with a process that is closely, really realistically, as close as you can get to simulating the conditions that make marine snow. These guys, this product is made and then preserved in that state. So when you feed it to corals, you get a really good feeding response, because they can actually taste it. They taste that it has actual live bacteria on it of a favorable type, and they will eat it. So if you don't use this stuff, it's pricier than some other foods you might use, but it doesn't necessarily have to be your primary food. You can use it as a supplement. So, for example, you can get your make sure that you're getting all the cobalamin, the vitamin B12 that you need. And you can also prime your corals with it. Put a little bit in the water, your corals will open up, and then they'll eat more of the coral feast or reef roids or whatever you put in there. So they're good in that way. And then you have the benefits that they will also, you're not just feeding the corals, but you're adding substances that copepods all the way down to certain types of beneficial bacteria need. So again, Yellowstone is kind of like a holistic supplemental food more than anything. And it was in fact developed alongside Probio to complement it. So it's very different from that product, but they kind of do, one does something that the other one doesn't do. So they're kind of hopefully balance each other out.
Right. Very cool. So that's something that's very fascinating about your products is, like you said, it's a holistic approach. It's very nutritious for your entire tank, and the Probio and the bacteria, the PNS bacteria in that is one that nitrifies and denitrifies and has all these different beneficial things about it that can help us in our reef tank. So I strongly want to encourage everyone to check out your website, and that's Hydrospace LLC.store, right?
Hydrospace.
Oh, Hydrospace.store. Yep. And then go over there and read all the blog posts about each product. And then they can also purchase your products at Algae Barn. That's where they're primarily sold, right?
That would probably be one of the better sources online. Yeah.
Okay.
You can buy it from our website, but it's the same price as Algae Barn. And at Algae Barn, they've got a really good shipping team, and then you can also get your pods and phyto and all that stuff in with the shipments. So And then lastly, we have a distributor in the United States, Aquarium Supply Distribution, ASD. So if you own a store, you can contact ASD to inquire about wholesale availability.
Okay, very cool. So touching back on denitrification, and like whenever we're learning about aquariums for the first time, we hear about beneficial bacteria, but we don't necessarily know what that means. And so this being able to look into what specific bacteria you're putting into your tank is very helpful. So is there any like final thought or final thing or advice or something that you would say about denitrification and your product and it balancing out your nitrate and phosphates and all that stuff in the reef tank?
Yeah. Maybe for the third time, it's worth saying, if you're thinking about denitrification or any other nitrogen transformation, think of it as an actual cycle where it's coming and going out of it throughout the tank, in and out of the atmosphere. And just know that you'll probably have to have all kinds of filter components and do other things. Reef tanks will never have the so-called slice of ocean in our living room. We're always going to have to play God and add a piece of equipment or a supplement or do an old-fashioned water change now and then. But think of it as a cycle and know that the bacteria in the tank will, beyond a doubt, have an influence on the cycle, because that's the base of the food system, of all the cycles, your carbon cycle, your phosphorus cycle. So be a little more mindful of that. In the future, I think it'll be easier to have some control over that. For one, hopefully manufacturers will start to identify, at least to the level of genus, the microbes that are included in their products, if not the culture methods and media. And then again, to throw a plug out for aquabiomics, aquabiomics.com. That is an incredibly useful tool that is right now underutilized. I would encourage everybody to try them out. If you don't know exactly what it is, you basically send a sample of your tank. I think one from the water column and one from like a swab, a surface. I send mine direct because I have a different way of doing things with them. But generally, they'll send you a kit, and then you take these samples and send it back to them, and then they analyze your tank, and then they'll tell you the relative abundance of different classes of microbes that are present in your tank, including the nitrifiers or other beneficial ones. And also, they'll let you know if you have any disease organisms that are present. So it's really interesting, but it's also quite useful because as we start, so he'll also send a report which kind of evaluates your particular unique microbiome and give you some suggestions as to how to improve your environment in the tank to promote the better types of bacteria, and maybe to promote a more natural community of bacteria and archaea in your system. But that's important because when people go to them and do this, they're basically building little tiny case studies. And what we'll find out over time is, well, what makes a great reef tank? And it may not necessarily be exactly like what's in the ocean. It's kind of like our reef tanks are analogous to greenhouses, right? Like we're not necessarily trying to recreate the tropical rainforest that this orchid grows in. We just want to focus on that orchid and make sure that we give it an optimal growing condition. And not, especially coral farmers, that's all they care about. They don't want a natural environment. They want an optimal environment. And a natural environment is never optimal. For organisms get by, they don't just thrive. That's how evolution works. But anyway, I would say, talk to Aquabiomics, give them their stuff, and let's build those case studies, and then we'll have an idea over time what works better in terms of a captive microbiome. And then if if more people are telling you what's in the bottle, we'll be able to actually act on that.
Yeah. And I just recently heard about them as well. And it's really interesting how specific they can get with the reporting back of what's in your water. And they also do the DNA test as well. So you got the microbiome, and the DNA. So it's pretty cool. Well, thank you so much, Ken, for joining us today on this episode and talking to us about denitrification and your product PNS Probio and your substrate sauce and your yellow snow and all that stuff. Great work, awesome products. So thank you so much for joining us.
You got it. Thanks for having me, and happy holidays.
Thank you so much for checking out this episode, and congratulations to Spidey Bry, who asked the question on Instagram that was featured in this episode. You've won yourself a PNS prize pack from Hydrospace. Be sure to check out PNS Probio, Substrate Sauce, and Yellow Snow from Hydrospace. Really great products and a lot of great work that Ken is doing over there. Thank you so much for all the support. Be sure to like and subscribe if you haven't already. If you have any questions, leave them in the comment sections below, and I'll see you next time on the Coral Reef Talk.